Cocrystal Pharma is a small but publicly traded company. This is our forward-looking statement. So today I'm going to focus on our drug discovery platform technology. In order to develop the broad spectrum, the antiviral therapeutics, we actually developed a platform approach which is unique in terms of the developing crystal pipeline first, and then identify the attractive drug binding pocket, which you will give the broad spectrum activity. And then the hit to lead process is a typical structure-based discovery process, and lead optimization is driven by PK study. So my talk would be 70%, I'm going to talk about the platform approach, and then 30% I'm going to show you the case study using PB2 lead molecule. So the Cocrystal, when we start the company, we decided to focus on viral replication enzymes. These enzymes are essential for viral genome replication and transcription. And we are currently looking at the several indications, the hepatitis C, as well as influenza and noro. We developed the other HRV, rhinovirus and dengue, using our platform approach. We developed the toolboxes as well. Most advanced molecule is the CC31244. We're currently conducting phase 2A study. This is a pan-genotypic HCV polymerase inhibitor. And then the second molecule is a novel helicase inhibitor. Both compounds are, again, pan-genotypic inhibitors with a higher barrier to drug resistance. Influenza, we have two programs, PA as well as a PB2. CC42344, I'm going to use this as a case study, how we built the pipeline. We're also working on the noro antiviral as well. So every technology or any platform has a pros and cons. And I think this is the one of the really challenging area. Everyone appreciates the high resolution X-ray data, but the problem is it takes a long time. And also, larger scale crystal production is really challenging. So this is just an example of our in-house Cocrystal, the capacities, and we're showing here. The HCV polymerase case, we have all of genotypes as well as drug resistance, and helicase, the same thing. Same thing with the influenza H1, N1, and H2, H3, H5, H7, and flu B, as well as the drug resistance crystals. The advantage of this platform approach, when you identify the hits, you will immediately know the binding site. And then also you can guide the chemistry. You can help the chemistry using this X-ray data. The panel A showing that 3 hits that binds the same pocket. If you overlay it, it's slightly different. And so if you show these pictures to chemists, then you immediately prioritize the chemistry. The panel B is showing the polymerase, HCV polymerase, the X-ray crystal structure. Replication enzymes have common properties. One, it recognizes, interacts with the nucleic acid, single-strand RNA or DNA. And also it has the very important protein-protein interaction modulate the catalytic activities. And if you're looking at here, slide B, for example, this particular inhibitor is blocking the RNA channel, not only blocking the catalysis. So you could actually use these all properties, design the molecule. And it's a very important approach. Panel C, we also define the broad spectrum activity by atomic level. We want to overlay all co-crystal structures. In this case, multiple genotypes you overlay. Everything together, you see the highly conserved binding mode. That's our broad spectrum activity definition. And overall, in order to execute this whole thing, you really have to have the larger scale without losing any X-ray resolution, larger scale crystal production, as well as overall data collection and computational work has to be done. So moving on to now influenza. So influenza, again, as a case study, I'm going to use a PB2 inhibitor. The top picture is showing here. This is all, again, the influenza crystals, H1N1, H2, H3, H5, H7. And we're going to just go through I'm going to walk you through the process. So again, I just want to remind you that influenza polymerase has a trimeric complex. It requires a PB1 and PB2 and PA. So if you look at the replication, always the first step is really critical, which is the PB2. It hijacks the host mRNA and brings the mRNA into the PA, which is the endonuclease active site, and then it generates the rapidly small primers. Once you have a primer, then polymerase will passively sort of initiate the replication. So PB2 is a really important step to control the replication process. There are several replication inhibitors already developed. Last two speakers present the baloxavir, which is a PA inhibitor. There is the first one, T705, favipiravir. And so I'm going to focus on, again, the PB2 cap binding inhibitors. So this, again, based on the known mechanism, there obviously this PB2 protein hijacks the mRNA. So all mRNA has this M7 GTP cap structures. So we and others confirmed that M7 GTP actually, in fact, that is a native ligand. PB2, you can see the PB2 domain, M7 GTP binds really well. Now, but wasn't really, validated as a drug target until we see the VX-787 was discovered. VX-787 was discovered based on high-throughput cell-based assay. The scientists at the Vertex, they identified the influenza compounds. They didn't know the drug target. When they did a drug resistance screening, they found the mutation is in all PB2 domain. So obviously it's targeting the PB2, and then they identified the binding site of the pimodivir. So when we start this, our starting point, again, selecting, once we develop the co-crystals and crystal pipeline, and the first thing is that we need to figure out whether this pocket is really is attractive for generating broad spectrum antivirals. So panel A is showing the sequence alignment. Obviously, it's highly conserved among influenza A strains. If you look at the panel B, we overlay all of our crystal structures, influenza A, and you can see the active site and the architecture is highly conserved. So once we have that, then we're also using different ligands to demonstrate the highly conserved binding mode. As you can see, using H1N1, we're just giving you some examples here. All using VX-787 compounds, they're showing the highly conserved binding mode. This is really important, and we have many different ligands demonstrate. So what that means is that this binding pocket is, we don't see any conformational change, any unusual binding mode. Again, the ligand binding mode is highly conserved. This is a really good starting point. It wasn't enough for us, so what we did is we want to try to figure out whether the PB2 biological function is hijacking mRNA, so M7 GTP information, that wasn't enough. So we actually screened the co-crystal structure with the RNA. So as you can see here, panel A, this is just assay development. Obviously PB2 has preference to a single-strand RNA over single-strand DNA. And then the panel B, we actually obtained the novel co-crystal structure with the single-strand RNA. What's important about this picture is when you look at the 5' end of the single-strand RNA, it's exactly overlay with the M7 GTP nucleotide. So this is a really important information for us to design the molecule, which way we want to move forward. So I'm going to just briefly go over. Once next step is how do you generate the hits? This is one of the approach that we use, fragment-based screening. Basically, we generate several thousands of crystals, and then we just basically use the cocktail. So typically 4 to 8 fragments per cocktail. We just soak with the compounds, and then ask a question: are there any ligands bound? So this is a non-biased. Crystal will pick the inhibitors. This is our property of our chemical libraries. It's diverse and lead-like, and just like anything else, in order to increase the hit rate, you really have to have an optimized assay condition. In this case, we spent a lot of time selecting the crystal form. In this case, H7N9 was the really best crystal form to increase the hit rate using the fragment-based screening. As you can see, panel A, the binding site is widely open. Panel B has the large solvent channels. We have a really successful fragment screening campaign. See, the panel A, I already showed you the binding pocket here is highly conserved. Our platform demonstrates that's the pocket that we should go after. Again, non-biased, without knowing any VX7A7 binding mode or anything else, you just ask a question: where is the hotspot in terms of starting point, right? And so panel B shows the cluster of the actual hit fragment is exactly binds the M7 GTP binding pocket. Again, this is non-biased hits, and you can see the fragments overlay structures is actually toward all hydrogen bonds or the molecular interaction is the same as M7 GTP. So this is a sort of foundation. That wasn't enough. So what we do is that we develop the drug-resistant crystal pipeline. Again, this is all these crystals. We typically we can grow up to 10,000 crystals per month. So we have this large-scale crystal production, and we map out each drug resistance. This is a in vitro screening, all published data. So we generate each drug resistant crystals and asking question: why this mutation caused the EC50, IC50 shift. So using the X-ray data, we map out and trying to understand the functional role of each amino acid residue, so we can work with the chemists to figure out what to do with the drug resistance. So just briefly, this is the our lead molecule. I'm going to just show you next a few minutes the property of compounds. 42344 binds the M7 GTP. Potency is a single-digit nanomolar. Showing good, based on preclinical characterization, showing good safety profile and the PK properties. Potency, just showing you the H1N1 pandemic strain 2009. Our compounds are 0.12 nanomolar. And just keep showing you the against other seasonal and pandemic strains, a single-digit nanomolar, as well as Tamiflu resistance strains. So all these list of the assays and studies are all done. At this point, the compounds are showing good PK data. This is, again, I'll be around after this talk, and if you have any questions, and I'll be happy to answer. But again, we looked at the cytotoxicity, mitochondrial toxicity, genotoxicity, cardiotoxicity, off-target, everything looks good. This is very initial PK data. The 45% have 57% the oral bioavailability, but now we have many different formulations. We can obtain up to 80% oral bioavailability. So moving on to the next topic here. Influenza case is provide a unique product opportunity, because influenza is virus targeting the lung, and you can actually develop the 3 different products. So we start with the oral route, but we're looking at inhalation and IV. Second one that we start is IV. Can we develop the formulation for IV? And then can we also develop the inhalation? So dry powder, initial goal was a dry powder, so we can actually complete the 2A study with the dry powder. And we were very lucky that we got the dry powder. The size particle is around 2 microns. So we did a quick PK study with the dry powder. This is a single-dose mouse study. Basically, you just introduce 1 puff. It's approximately, the powder is 0.8 to 1.2 milligram. The drug concentration approximately 200 microgram. And this is a single dose, and then we're doing the PK. PK study. You can see that 1 hour, and the compounds immediately goes along up to 1 millimolar, almost 1 millimolar. At the same time, if you measure the plasma concentration, is 5.6 micromolar. And 24 hours, the same thing. We just trace the compounds. So we're really excited about this data. And typically, the compounds works for oral routes, and it doesn't work for physical chemical property. Doesn't work for other routes. But we're lucky. This compound is showing the very interesting dry powder. Now, drug resistance, this is an ongoing study, so I just want to share the current data. So we screened this side by side with the VX-787, our compound. So far, VX-787, we identified the 3 drug resistance. Published data was a 7, but so far we found the 3. Our compounds, we found the 1. Even though binding pocket is the same, if you look at the residues, completely different. Very interesting. So I'm going to just briefly show you the movie before I show you discuss our hypothesis. So influenza polymerase complex is a very dynamic molecule. If you look at the blue, that's a PB2. So PB2 actually is a kind of like a cowboy whip. It takes mRNA and put it in the PA active site. So this compound again, PB2 is rotates. And when we measure with the activity against the purified PA endonuclease, VX-787 as well as our compounds, and there is no inhibitory activity, right? So then we measure the compound with the trimeric complex. Remember, the influenza polymerase actual complex is a trimeric. We assay them, and the VX-787 does not inhibit the PA, endonuclease activity. Our compounds does. So again, our current hypothesis is that in solution, the PB2, when you have substrate, it rotates. Even though binding pocket is the same, our compounds actually, prevents the rotating of the PB2 in addition to competing the mRNA. So which is pretty interesting. So we want to look at the in vitro combination effects. So we tested with the 3 different drugs here, Baloxavir, as well as favipiravir and Tamiflu, and our compounds are showing strong synergistic effects with all 3 compounds. Just a quick summary. Again, our platform approach. We start with the crystal information, high-resolution crystal information. Not only one serotype, we look at all of the serotypes, and that's a really powerful approach to look at the protein-protein interactions and ligand interactions as well. So using that platform approach, and then we developed the antiviral broad spectrum, antiviral single-digit animal compounds, again, drug resistance profile I mentioned to you, and as well as we hope to develop the multiple products with this lead molecule. Chemical stability is outstanding, 12 months, and has the synergistic effects. I want to thank my team and collaborators and other supporters. Thank you for your attention.